Okay, so when we're doing genomics, the sequencers just flood us with this ocean of raw data. It's chaotic. So how do we possibly go from all that noise to finding one single, meaningful genetic variant? Well, today we're going to walk through the absolute gold standard for doing exactly that, the GATK best practices for germline discovery. To really get your head around the scale of this, just imagine this for a second. You have to find one tiny typo in a book that has 3 billion letters. That's what we're up against when we hunt for genetic variants. And it's not just about finding a needle in a haystack, it's about being 100% sure that what you found is actually a needle and not just, a weird-looking piece of hay. So, how on earth do we do that? Well, the answer is a really, really rigorous step-by-step process. So here's our plan. First, we'll get a bird's eye view of the whole path. Then, we're going to dive deep into Phase 1, pre-processing the data. After that, we'll tackle Phase 2, actually calling the variants. And we'll wrap it all up by looking at the final prize, the VCF file.
All right, let's get started. Section 1, the path to discovery. Think of this as us laying out the map for our entire journey.
And here it is. This is our map, the GATK best practices workflow. You can see how it just flows from left to right, right? It takes that messy, raw sequencing data and just systematically cleans it, refines it, and polishes it until we're left with these super clean, high-confidence results. This is going to be our guide for the rest of this explainer. So every journey has a starting point, and for us, it's something called a FASTQ file. The best way to think of this is like getting millions of tiny, shredded sentences that have been ripped out of that giant 3 billion letter book. This is the raw stuff, straight off the sequencing machine. And it doesn't just have the genetic reads, it also have these quality scores for every single letter, which is basically the machine telling us how confident it is about what it saw.
Okay, that brings us to Phase 1, pre-processing. This whole stage is about forging a clean path forward from all that raw data. I mean, you can't find real genetic variants in messy data any more than you can build a solid house on a shaky foundation. This part is just absolutely crucial.
So this whole pre-processing thing really boils down to 3 key steps. First, we have to align all of our reads to a reference genome. Second, we need to hunt down and mark any duplicate reads. And third, we do this really clever thing called base quality score recalibration. Let's break each of those down. So step 1 is all about taking those millions of tiny shredded reads and mapping them back to a known reference genome. It's kind of like taking all those ripped up sentences and carefully pasting them back into a master copy of the book right where they belong. This gives us our first really structured file, which is called a BAM file. Next up, we have to mark duplicates. See, back in the lab when the DNA is being prepped, there's a step called PCR amplification, where some bits of DNA can get copied over and over and over again. These aren't unique biological insights, they're just technical artifacts. So this step is all about finding those copies and flagging them so they don't mess up our results. So why is this such a big deal? Well, imagine you see 10 copies of a read, and they all have the same typo. You'd probably think, wow, that's got to be a real variant. But what if all 10 of those copies came from just one original piece of DNA? Then it's not 10 pieces of evidence, it's just one piece that's been artificially amplified. Marking the duplicates stops this kind of false evidence from tricking us.
And now for the really cool part. Base Quality Score Recalibration, or BQSR. This is, believe it or not, a machine learning step. Sequencing machines are amazing, but they're not perfect. They have their own little quirks and make systematic errors. What BQSR does is it builds a model of those specific errors and then goes back and adjusts the quality scores for every single base in our data.
I mean, think about that. This step is essentially teaching the machine how to correct its own mistakes, and doing this dramatically improves the accuracy of everything that comes next. It's our final cleanup, making sure our data is as pristine as possible.
Okay, so with our data all cleaned up and perfectly prepped, we are finally ready for Phase 2, calling variants. This is the fun part. This is where we stop cleaning and start actually discovering the genetic differences we've been looking for. Now this phase is all about turning potential signals into high-confidence results, and we do that with 2 main processes. First, we do the actual variant calling using a tool called haplotype caller. And second, we apply a final, rigorous round of filtering. So what in the world is haplotype caller? Well, this is the powerhouse, the core engine of discovery. It goes through our beautiful, clean reads and performs what's called a local de novo assembly. Basically, it reconstructs tiny little sections of the genome from scratch. And by doing that, it can spot even the tiniest differences, like single letter changes, which we call SNPs, or even little insertions or deletions called indels. Now, the real magic here is how it does this. Haplotype caller doesn't just look at one read at a time like a simple scanner. No, it looks at all the reads in a small area together. And then it asks a much smarter question. What is the most likely original sequence that would have produced all of these different reads I'm seeing? It's this group-based logic that makes it so incredibly accurate.
Now, even after a haplotype caller gives us a list of potential variants, we're still not quite done. The last step in this phase is final filtering. This is our last major quality control checkpoint. We apply some really strict statistical filters to separate the true biological variants from any leftover noise or technical junk.
The goal here is simple, but it is so important. We want to throw out the artifacts and keep only the high-quality variants. This ensures that what we end up with has the absolute highest probability of being biologically real.
And that, finally, brings us to our destination, the final VCF file. This is where we get to see the fruits of all our labor, reliable biological insights.
The final output, the treasure at the end of this whole journey, is a VCF file. VCF stands for Variant Call Format. It's a simple, standardized text file that just neatly lists out all of the high-confidence variants that we discovered, along with a ton of useful information about each one. And here's a super simplified peek at what's inside. It tells you the chromosome, the exact position, and what kind of variant we found there. A SNP, an indel, you name it. This is it. The final, organized list of every single typo we confidently found in that 3 billion-letter book. And this file is the key. It's what unlocks all the amazing downstream biological analysis. Whether a scientist is studying disease, or evolution, or population genetics, this high-quality VCF is where their new discoveries begin. So, let's just quickly recap the big takeaways. First, you absolutely need a structured path like this to get reliable results. Second, all that data pre-processing isn't just busy work, it is the foundation for everything. Third, remember that variant calling finds the candidates, but it's the filtering that refines them into truth. And finally, that all-important VCF file is what gives us the high-confidence results we need to make new discoveries.
So that really just leaves us with one final thought. Now that we have this incredibly powerful and standardized map to navigate our way through all this genomic data, what new biological discoveries are now within our reach?